Okay, so now we come to the applications of CRISPR and how it can be used to modify the genomes of all types of cells, including mammalian cells. So it can be used to address basic biomedical questions, just like an RNAi, RNA interference experiment, which I showed 2 weeks ago, by adding in a guide strand which is complementary to the target genomic sequence. You can create a double-strand break. And here you can see a target single guide RNA which has been introduced into a mammalian cell, or a control single guide, because when you're doing experiments like this you also need to control for it. You need to have a single guide RNA which would not be predicted to target any region of the genome. And this is just an example. So then when you do a Western blot, for example, for your target, you'll see that the control single guide RNA is not affected, the target sequence. But when you add your target sequence in here into cells in parallel, that gene has been will have been a double strand break, and it won't be transcribed and translated into protein, so you don't see it on your Western blot. But there is protein there, because when you do a Western blot for GAPDH, an unrelated protein, you can see that there's protein in both wells. So you've specifically knocked out that particular gene. And another basic biomedical question you can address is genome-wide screening. Because now we know all the genes in the human genome, there's about 20,000 of them. You can create a guide that targets each one of those genes individually. And so you can buy in a CRISPR library, whereby in 96 well plates, whereas each well of the 96 well plate will contain a guide strand that targets your particular gene of interest. So if you put your cells in there, and you can get your Cas9 gene in there as well. So you could get your guide strand and your Cas9 gene into the cells by electroporation, for example, by applying electricity to the cells. So in each well then you'll have knocked out a particular gene, and you'll know which gene it is because you'll have a data sheet showing you that what guide strand is in there, what gene it targets. So this guide strand could target actin, this gene here could target, for example, the T cell receptor. And with that then you can stimulate your cells, for example, to produce cytokines, and then you could see which genes you've knocked out have led to a reduction in the cytokine levels. Or example, if these were cancer cells, you could add a guide strand in here, knock out the different genes and find out which cells have died by doing a proliferation assay. So with that you can identify hits, and so you could identify basic biomedical questions: what genes are involved in a particular process, for example, producing cytokines in T cells, or which ones are required for survival for cancer cells, and identify new genes, because we know we have 20,000 genes, but we don't know what they all do in terms of individual cells. And this shows how people have got on board the CRISPR train in the last few years. CRISPR, the word CRISPR, although it was discovered in 1993, it wasn't called CRISPR at the time. It was only a few years later when that term CRISPR was first agreed on by several research groups. So the first time CRISPR appeared in the PubMed literature was 2002, with just one publication, and up to 2007. When Philip Horvath's paper came out, there's only 12 publications at that stage on CRISPR, even though these repeat sequences were long known back as far as 1993. And since 2013 and 2014, you're now seeing proliferation of up to 500 articles since then. Really, up to last year, we had almost 4,000 articles on CRISPR. So it's been a massively exploding field, whereby many labs, even in the last few years, obviously have started to publish results on CRISPR and knocking out genes by using CRISPR technology rather than relying on other things like RNA interference, whereby RNA interference is you just knock down a gene. You're not going to completely knock it out, because you might only knock down the gene by 80%. You might suppress that messenger RNA being translated to protein by 80%, so you still may have 20% of the protein there. Whereas with CRISPR, you can completely knock it out, so you can completely remove it from your cells. So it's a much more specific reagent. In 2019, there's been a lot of publications to date as well, even since March now, with 1,500 publications. So it's going up and up, where people are using it much more often in their biomedical research. And of course, industry have got on board as well, because we can already produce genetically modified foods. We can produce genetically modified plants, for example. There's been genetically modified apples which don't bruise anymore. They have genes inserted into them There's plants now which are on the market which are resistant to insects and resistant to weed killers. And we also have animals, genetically modified animals now, whereby genes have been inserted into these animals to produce biopharmaceuticals or to make salmon which grow quicker. So it's obvious that CRISPR is actually been widely used on these as well, to genetically modify specific areas of the genome for similar processes. So to make plants more resistant to pesticides and make them grow quicker and give a higher yield, and even to produce genetically modified animals for pharmaceuticals. But none of these have reached the market as such yet, although there are some genetically modified foods which are using CRISPR. We'll see those coming on board more and more. And that's for food and plants. But what about therapeutics then? That's obviously the key area, applications of CRISPR. Can you use CRISPR technology for therapeutics to genetically modify the genomes of humans? And as I'm going back to my first couple of slides, where there's many ways you can do that. You can modify the genes of humans through antisense or RNA interference. You can block the expression of a gene, and there's a number of clinically approved drugs for that on the market. There's a number of gene therapies now where you can replace genes, missing or defective genes, using a viral vector to get that gene into host cells. And with gene editing, zinc fingers and talons have not been clinically approved, and with CRISPR being seems to be much more efficient at creating double-strand breaks in DNA. It's certainly going to be one which is more widely used. No clinical approvals yet for CRISPR technology, but there's certainly I think we're going to see that in the next couple of years. Some of the potential of CRISPR applications in curing diseases. 2 papers appeared in Science back to back just over 2 years ago, whereby in mice who have the mouse version of muscular dystrophy. In humans, muscular dystrophy is caused, it's a muscle wasting condition caused by a genetic defect in the dystrophin gene. It's a very, very big, long gene, and there's multiple types of mutations. But essentially, patients with muscular dystrophy have a mutation. They inherit this mutation in the dystrophin gene so that they don't produce a dystrophin protein. So scientists, Eric Olson and Charles Garbash, 2 independent scientists, published these papers back to back, whereby in mice who have the mutant dystrophin gene, they could use CRISPR technology to essentially remove that mutation in these mice. These weren't done and basically replaced that defective gene with the gene edited form and replaced the dystrophin mutation. And with these mice, these weren't given to embryonic mice, these were given to living mice, these were given to adult mice. So the CRISPR system was used encoded in a viral vector, actually. So they used the adeno-associated virus here. That viral vector, which had in its genome, the viral vector, the Cas9 gene and the guide strand encoded on that. That goes into the muscle of the mice, and the CRISPR does its thing. It uses the guide strand to target the particular part of the dystrophin gene, creates a double strand break, and then you get repair of the dystrophin gene. And in these mice, then, if you read the article, each of these methods restored dystrophin protein expression in cardiac and skeletal muscle to varying degrees, and expression increased from 3 to 12 weeks after injection. Postnatal gene editing also enhanced skeletal muscle function, as measured by grip strength tests 4 weeks after injection. So the mice were able to regain their muscle function, which is amazing. And in the similar paper back to back, there's a deletion in exon 23 of the dystrophin gene that was altered by the CRISPR-Cas9 system. Partial recovery of functional dystrophin protein in skeletal fibers and cardiac muscle, improvement of muscle biochemistry, and significant enhancement of muscle force. So you don't have to use this in the embryonic stage. So you don't have to use it on an embryo of a human. Potentially, if they have the inherited form of it, you could actually give it to a child, for example, to use CRISPR. Once you can get the Cas9 and CRISPR system to the right area, particularly using a viral vector. So with that, we've seen that was only done in animals, so can it be done in humans? And the first clinical trial has just gone ahead. It was started in September 2018, so just under 6 months ago. It was granted by the FDA, and it's an ex vivo CRISPR clinical trial in humans. So what it has done is to genetically modify T cells to make them target cancer. And just to reiterate what T cells are again, so their T cell receptor is comprised of the alpha and beta chain, which arise by recombination. where the portions up here get genetically rearranged. And this alpha beta chain then recognizes a peptide antigen, which is presented to it from an antigen presenting cell. And then the second T cell, which will be produced in the bone marrow, because its alpha and beta chains will undergo a different genetic rearrangement, they'll recognize a different peptide antigen. So in our bodies then we have billions of T cells and they all recognize different antigens. Some will be bacterial antigens they recognize, some will be self antigens. Others will be potentially cancer antigens. So what the scientists have done in Philadelphia is they're proposing in their clinical trial is they're going to take T cells from patients and they're going to genetically express a new T cell receptor, a new alpha and beta chain for this peptide antigen, the NY-ESO antigen, which is highly expressed in melanoma cells, sarcoma cells, and multiple myeloma tumor cells. So with that, then they're taking the patients all of their T cells, which will all recognize different antigens, but redirecting them to express this new alpha beta chain, shown here and here. A new alpha beta chain on their surface here, and on their second T cell, a new alpha beta chain, expressing this cancer antigen. So what they'll do is they'll inject those back into the person's body, and what will happen is those T cells should target the NY-ESO antigen, which will be expressed on the melanoma cells or the sarcoma cells and multiple myeloma tumors. Now, the only issue is when you express a new alpha and beta chain in a T cell, there's the potential, and has been shown in animals, that the alpha chain of the newly introduced T cell receptor, this alpha chain here is specific for the NY-ESO antigen, could potentially pair with the endogenous beta chain that was already there on the T cell as it developed. So, for example, this is specific for E. coli, this alpha beta chain. This beta chain here for the tumor antigen could pair with the alpha chain for the E. coli. So you've created a new T cell receptor, which you don't want to, and that could go and cross-react with something in the body like a self-tissue, which has been shown in animals and lead to death of the animals. So you don't want that. So what they're using in their trial is they're going to use Crispr, so that as well as expressing the alpha beta chain for the tumor antigen, they're going to use CRISPR to genetically create a double strand break in the alpha and beta chain of the endogenous T cell receptor. So they're knocking this out here of all the patient's T cells. So all their T cells now that they've taken from their blood will only have the alpha and beta chain to the NY-ESO antigen. And they've also introduced another guide strand in there, because you can introduce several guide strands on your construct. And they are using a CRISPR guide strand to knock out the PD-1 inhibitory receptor. Because T cells, when they get into tumors, one of the way the tumor switches off a T cell is it expresses a protein called PD ligand 1, and that targets the PD-1 receptor on T cells and makes it become dysfunctional. It becomes anergic. It doesn't signal anymore. So what they've done, if they've knocked out on the T cells as well, all the patient's T cells, the PD-1 inhibitory receptor, so that makes the T cells switched on as well. So that's their proposal, that they're going to infuse these modified T cells. So they're modified different ways. They have a new T cell receptor, and they've lost the endogenous receptor as well as the PD-1 receptor. They're all gone. Going to put those back into the patients and see what is the tumor response. Will these patients who have these three types of tumors respond to them? So that's going to be interesting. That trial has just been underway, been enrolling since last September. It's a phase 1 trial, so we'll see. Hopefully there's no toxicity. That's what the phase 1 trial is proposed to do, look at toxicity in the patients with these transduced T cells and hopefully see a reduction in tumour volume. That's the first clinically approved clinical trial for use by the FDA. That was ex vivo, so that means the CRISPR has been used on the cells, for the patient cells, in vitro. But just soon after, last November, the FDA gave the go-ahead for the first in vivo CRISPR clinical trial in humans in the USA. And this is to treat a form of congenital blindness. So this CRISPR gene editing there's a condition, an inherited condition known as Leber's congenital amaurosis type 10. And it's the most common form of inherited childhood blindness, where children can be born with this condition. They've inherited a mutated gene, and that they have very poor vision, and as they progress through life, or even before adolescence, they can lose their sight altogether. So the idea is to target that mutation in these children. And obviously, you want to target that mutation. You can't take their eye out, you can't do it ex vivo, so you have to get the CRISPR system into the child's eye, the affected person's eye. And the gene in question is CEP290. There's a particular mutation encoding in that gene where you can target with the CRISPR. So you just introduce with your Cas9 enzyme the guide strand, which will hybridize to the CEP290 gene and guide the Cas9 to that. Now, you could ask the question, because this gene, the CEP290 gene, is just mutated, could you not just do gene therapy? Because just last year, the FDA introduced the first gene therapy, in vivo gene therapy, Luxturna, whereby on an adeno-associated virus, they've encoded a new gene for RPE65, and that's injected. The adeno-associated virus encoding the new RPE gene is just injected into the retinal cells here to express the new gene. So could you not just express the CEP290 gene, like here in the adeno-associated virus, into the retinal cells as well for these patients? But you can't, because the CEP290 gene is very, very big. Remember last week I mentioned the adeno-associated viruses can only take an insert of about 5 kilobases. So the CEP290 gene is much, much larger. So you couldn't just give that CEP290 gene back into these patients to restore the gene. So the best way to do that is probably through CRISPR, whereby you can specifically target that mutation through the Cas9 enzyme. So Editas is the pharmaceutical company, a new CRISPR company which was founded by Jennifer Doudna and others. They've created this guide strand called EDIT101, and it's a CRISPR system, and it's encoded on an adeno-associated viral vector. So that's encoded in the genome of the adeno-associated viral vector, which is non-integrating, so it doesn't integrate into the genome of the patient. That's going to be injected into the retinal cells here, and the CRISPR system then will be expressed by the adeno-associated viral vector. You get your Cas9 being expressed, and your guide strand being expressed. And with that then the guide strand will target the Cas9 to that CEP290 gene. And the idea is then it'll restore that genetic mutation in these patients. It's a phase 1 clinical trial. Hasn't started yet, it's recruiting in June. The first patients are expected to be recruited in June. So hopefully maybe that will come to fruition. We'll see hopefully if they can restore sight to or stop the progression of blindness in these patients. So of course, all these CRISPR gene therapies which have been done so far have been done in somatic cells, so non-sex cells. So like muscular dystrophy in dogs has been done, and mice, as I mentioned. That's just giving it directly to the muscle cells. And as well as the blindness trial here, the CEP290 gene, that's done in the eye cells. So that genetic mutation is not in all the person cells. But with CRISPR, there's the potential, can you use it as a genomic editing tool on embryos? Could you create embryos whereby each cell in the body has been genetically modified through CRISPR? And that has actually just been realized in the last couple of months. Because potentially what you can do is, if you have a fertilized egg, which contains the DNA from the sperm and the egg, or you can genetically modify a nuclei from either the sperm alone or the egg cell. If you know, for example, the father was carrying a genetic mutation, you could potentially use the CRISPR-Cas9 system to correct that mutation, and then do in vitro fertilization, so that you'll know that that's now that DNA has now been corrected, and you get your fertilized egg. And when that egg develops into 2 cells, 4 cells, and so on, each of those cells will have the new genetic rearrangement have been brought about by CRISPR-Cas9, so that you could potentially have an offspring then in all of its cells have been modified through CRISPR-Cas9. But the issue with that is that's creating a designer baby, because that baby's cells, all of their somatic cells, their sex cells and their non-sex cells, will be genetically modified through Cas9. So if they produce offspring later on, they're going to pass on that gene-edited genome to their offspring. So that creates a massive ethical debate. People often think, is this just an ethical debate? It's never going to happen. A paper published in Nature just a year and a half ago, whereby they didn't didn't allow these embryos to progress to full fetuses, but they showed that you could correct a pathogenic gene mutation in human embryos. This was from a group, a multi-international group, and they could use CRISPR-Cas9 to correct a genetic mutation in an embryo. Obviously, that's very good. You can cure obviously inherited diseases. There's the potential for that very specifically. This mutation they changed was the MYBPC3 mutation, and they could use precise CRISPR-Cas9 based targeting to change that mutation and to remove that mutation in these embryos. Now, they didn't allow these to go to full fruition. And that was the major advance really of this, is showing that the potential that you can genetically modify a human using Cas9. But however, just a few months ago, November 2018, it was to much consternation around the world, the Chinese geneticist Jiang Kui, he announced over in China that he had created the world's first gene-edited babies using CRISPR. This is him when he announced it. So just a couple of months ago, what he did was he recruited couples and who had either the dads had HIV, and obviously when a father conceives a child with a mother and the father has HIV, that HIV virus can be passed to the offspring through the mother's placenta. So what he was trying to do, and what he claimed to do, was that one of the ways the HIV virus gets into cells is through the CCR5, the chemokine receptor 5 on target cells. So he wanted to use CRISPR technology to knock out the CCR5 chemokines. So what he did was he got embryos, which he generated through IVF. So the fathers had HIV. He took their sperm and he took the mother's eggs. He did in vitro fertilization. And thereafter, he used CRISPR-Cas9 to knock out the CCR5 chemokine on the embryos. So those embryos, which were divided in 2 and divided in 4, divided in 8, they all were missing the CCR5 chemokine, which he has alleged, because he hasn't published the paper yet, he just announced this. What he did then was, which created a major ethical debate, was he inserted those genetically modified embryos back into the women. He implanted those embryos back into the women. It was one mother so far, and she gave birth to twin girls. So those twin girls allegedly are now alive, healthy, but they're missing the CCR5 chemokine receptor. And he said they're free of HIV. So that came to major consternation around the world. First of all, in terms of ethics, because he said he had ethical approval from his host institution. Subsequently found that he made it up. He didn't get ethical approval at all. There's debate whether what he's said is true at all, because he hasn't published it or no one has seen the data. But he's been fired from his job in the last few weeks actually. So he's no longer working there. So it has created that potentially you can cure diseases, but you can also create. People are afraid you can create a monster. Where is this going to lead to? So come a long way really from 1993, just showing these repeat sequences and spacer sequences and being able to do this genetic engineering by introducing CRISPR-Cas9 into human cells. And there's been a major war ongoing since CRISPR has come to the fore, particularly from the scientists who were really key in getting CRISPR from an in vitro work into mammalian cells, showing it can be used to edit the genomes of mammalian cells. George Church at Harvard, Feng Zhang at Broad Institute, Jennifer Doudna at University of California, and Emmanuel Charpentier, they did collaborate together. They're no longer working together. There's been a major war because, first of all, there's been a patent dispute as to who discovered CRISPR and who owns the technology, because obviously it can be used for a huge range of diseases. These are some of the diseases like modifying T cells, modifying stem cells, knocking out mutations such as muscular dystrophy, curing cystic fibrosis potentially, creating new livestock, industrial biology, agriculture, plants and animals, drug discovery, research tools, and in the lab using them as a research tool. So the people who have filed saying that they own it is Emmanuelle Charpentier. She said she was one of the first people to file a patent on CRISPR. Whereas University of California, Berkeley, where Jennifer Doudna works, she's filed a patent, as well as Feng Zhang at the Broad Institute. Now, there's been a major debate as to who filed their patent first. Doudna filed hers patent first, seemingly, but Feng Zhang submitted his afterwards. but he used an expedited mechanism to get his looked at first. So there's been a major debate between California and broad institutions to who owns CRISPR technology, because if you want to use it, then you'd have to pay them to use it, and it's obviously going to be a multi-billion-dollar industry. So there's been a major war. What was happened is Dudna's patent seemingly said it could be used for genome editing, but she didn't specifically mention humans, whereas Feng's patent said it could be used for genome editing in humans, and he was awarded the patent. There's been counterclaims from Jennifer Doudna because she said it was obvious that this could be used in humans, but in fact the patent actually didn't state it. So many of these scientists are linked with different companies. Doudna has set up 2 companies, Intelia Therapeutics and Caribou Sciences. I mentioned she used to work with Emmanuel Charpentier in that paper, where they first showed that you could make a guide strand rather than using the tracer RNA and the CRISPR RNA together. You can just use a guide strand to guide the Cas9 to the target sequence. They no longer work together, they've gone their separate ways. Carpentier has made her own companies, ERS Genomics and CRISPR Therapeutics. If you follow the lines, you can see what diseases their companies are primarily targeting. So there's been a lot of war going on between all these scientists and primarily the universities where they're from, where they're working, as to who owns the technology. So it's back and forth. At the minute, Zhang has the patent, as far as I know, where there's currently a counterclaim put in by Doudna at University of California. So it's been getting nasty. So obviously it's major technology, and it's shown that CRISPR started out from very humble beginnings from an innocuous publication really in 1993 by the Spanish scientist, just showing these repeat sequences in bacteria and archaea. But it's widely accepted as one of the most exciting gene therapies, giant gene editing technologies that's going to provide major advances both in biomedical research and the development of new therapies. Really is precision medicine because you can target a base, a single nucleotide, anywhere potentially in the genome to correct a mutation or to insert a new gene or to remove a gene. And there's clinical trials underway. On the clinicaltrials.gov website in the States, there's 26 CRISPR trials undergoing now. I mentioned there's one in vivo and one ex vivo, but there's many more have started as well. Obviously, these are mainly in the States. You're not allowed to work on embryos in the States, do embryonic research. But obviously in China, we've seen that has been flouted by the scientists over there went ahead and just genetically modified embryos themselves, created humans from that allegedly. So there's major ethical concerns regarding the manipulation of sex cells and embryos for that. One of the major issues of CRISPR is potential off-target effects. It's widely accepted that CRISPR is very specific in that your guide strand guides your Cas9 to your genomic region of interest. But how specific is that? Because there has been shown that your guide strand can guide Cas9 to the site you want, but it can also target it to other sites as well. Even though there's not 100% sequence homology between your guide strand and your genome sequence, it can cause Cas9 to create double strand breaks at other regions of the genome, and these are known as off-target effects. So that's one of the major issues with the field. But as mentioned, Cas9 has been modified in different ways to change its function and make it more specific. So people are showing that scientists are now showing new publications coming through all the time that you can get better Cas9s being produced to modify the genome more specifically and not create these off-target effects. So went from very humble beginnings to very exciting area, with obviously ethical concerns as well. So that's it. So that's CRISPR in a nutshell.